Welcome, everyone. We'll give it just a minute or two for everyone to join the webinar, and then we'll get started. Morning, everyone. Yeah, we'll give it probably one more minute or so. This is really fun. We have people say hi and say where they're joining from. So if you want to pop that in the chat, go for it. University of Michigan, Vanderbilt. Mike said hi. Oh, Xavier, love to see it. Looks like your squad is strong. You got a lot of supportive messages.
Hope so.
Yeah, lots of people from Xavier. All right, we will go ahead and get started. It's so great to see people joining from all over. That's just warms my heart. All right, welcome everyone. I am Brielle Ferguson. I am a co-founder and director of programs for Black in Neuro, and I'm so excited to welcome you today to our last Black in Neuro seminar of the year with the amazing Dr. Austin Coley. Just a couple things before we get started. Before we begin, I would like to acknowledge that I am joining you today from Muwekma, Ohlone, and Ramaytush lands. We were all joining this event from different places around the world, so I invite you to take a moment to reflect on the lands you're joining from, as well as your relationship to those lands and the indigenous people who have and continue to inhabit them. Another quick technical note, if you would like to access the live transcript, that's available down through, if you click on the three dots at the bottom right corner of the screen. Also, if you'd like to share any tidbits about anything you learned today, you can do so on social media using the hashtag #SpinSeminarSeries. And in terms of flow for today, Austin will present for about 45 minutes, and then we'll have some Q&A at the end. If you have questions, feel free to drop them in the Q&A function, and I'll ask them live during the Q&A portion. Okay, so I am super, super excited to introduce Austin Coley. There's just so much I could say about him. This just brings me so much joy. He did his bachelor in science, or got his bachelor in science from North Carolina Central in biology. He went on to get his master's from Case Western in cell physiology. He then went on to do his PhD at Drexel University College of Medicine, where he and I actually worked together in Dr. Wenjun Gao's lab. His PhD work involved investigating the role of PSC95 in synaptic transmission, all the way from the protein and physiology level up to the level of behavior. There he won several both internal and external awards for his research and his service contributions, and was part of, I believe, the inaugural cohort of the prestigious F99 K00 award for his dissertation work. He completed his PhD in 2019 and then went on to join K-Tai's lab at the Salk Institute. There he studies how neural circuits regulate behavior and state-dependent region-specific cellular aberrations that are implicated in a range of neuropsychiatric disorders. To test this, he uses multiple different techniques, including in vivo 2-photon calcium imaging, ex vivo whole cell patch clamp electrophysiology, to get a hold of ensembles that are important in synaptic plasticity in an anhedonic mouse model. Outside of the lab, he is incredibly active in the community and regularly gives career seminars seminars from grade school all the way up to the undergrad level to help change and reframe the image of what a scientist looks like. More recently, as Salk, he founded the Black Association, which he also chairs. So as you can see, he does a lot inside and outside the lab. So we're super lucky to have him here. Today, he's going to talk about his new work, and I can't wait to hear about it. So without further ado, I will pass it off to Austin.
So thank you for that amazing introduction, Brielle. I'm very excited to give this talk for the Black and Neuro fam. I'm an extreme fan of this organization, and also I'm an extreme fan of my host for today, Dr. Brielle Ferguson, who we share this similar lineage with, coming out of Dr. Wen-Jun Gao's laboratory at Drexel University. So I'm very excited to give this talk today to discuss my research as well as my activities outside of the laboratory. So the title of my project is Investigating Medial Prefrontal Cortex Valence-Specific Neuronal Populations During Anedonia. So mental health illnesses can alter our everyday life. It can alter our motivation. It can affect our attention and focus. And it could even affect our social interactions. And the DSM, which stands for the Diagnostic and Statistical Manual of Mental Disorders, is used by healthcare professionals to classify these mental illnesses. However, it transitioned more from this categorical approach to more this dimensional approach due to the convoluted spectrum of symptoms within these disorders. And this makes it difficult for treatment intervention. So a key question of my research is, how do we differentiate amongst these neuropsychiatric disorders? And I'll give you this key example of these two disorders. First, major depressive disorder, when you're thinking more of the canonical features, such as sleep disturbance, depressed mood, feelings of worthlessness, also suicidal thoughts, agitation, and fatigue. In comparison to schizophrenia, where you're probably more so thinking of just the positive symptoms, such as delusions and hallucinations, but there's also cognitive symptoms, such as impaired working memory, impaired executive function, and also the negative symptoms that's associated with it, such as a lack of motivation, disorganized thoughts, and even emotional blunting. If you notice that within these two disorders, there's an overlap of conditions or symptoms, and that's anedonia and inattentiveness. And in fact, 25% of people diagnosed with schizophrenia, they meet the criteria for depression. And anedonia is suggested to be a vulnerability marker. For example, within schizophrenic patients, before they start to experience their psychotic symptoms, during the prodromal phase, this is the early onset phase, this is where these patients start to experience their depressive-like phenotypes, and anedonia is one of them. So a key question in my research is, how do we isolate these condition-specific changes in neuropsychiatric disorders? So I'll first give you a preview of what we do in Dr. Kay Tai's laboratory at the Salk Institute. We're primarily responsible for investigating these valence processing and state-dependent changes. And valence processing can be used to describe the emotional state by assigning the hedonic value, whether it be positive or negative. And this could further describe the motivation to seek rewards or the motivation to avoid punishment. And what we're primarily responsible for are investigating the neuronal populations as well as the neural circuits that are associated with valence processing. And one of the key regions that we're focusing on is the medial prefrontal cortex. So the prefrontal cortex is primarily responsible for cognitive processes such as learning and working memory, but it's also responsible for sociability and even emotional processing. So this is a review that we published earlier this year where we discussed the dynamic abilities of the prefrontal cortex. And in one of the chapters, we focus on reward processing. We look at specific functions such as reward anticipation, reward consumption, and reward prediction, and even aversion, and even anedonia. So we highlight the specific circuits, whether it be downstream regions of the prefrontal cortex or the specific inputs into the prefrontal cortex. And one of the circuits that I want to discuss today is the VTA circuit to the prefrontal cortex. Although it's very sparse, it is responsible for negatively processing this valence stimuli. And one key example that I want to give you today was done by Caitlin Vanderweel, a previous PhD student, within the Thai laboratory, and she used in vivo electrophysiology as well as optogenetics to selectively photostimulate these dopaminergic neurons within the prefrontal cortex, while simultaneously recording from the prefrontal cortex to these DPAG neurons within the brainstem. And what she showed was that in response to specific stimuli, an air puff, which is used as an aversive stimulus, that there's an increase in the signal to noise ratio within those specific circuits that was not shown in response to sucrose, which is a reward stimulus. So this led to a key question of my research is, how does dopamine modulation alter the prefrontal cortex projection-defined pathways during these neuropsychiatric conditions? And the first thing we wanted to look at was a previous literature discussing the prefrontal cortex circuits that are implicated in depressive-like phenotypes. So what is known about major depressive disorder is that there is a dysregulation in prefrontal cortex neural activity. And this was shown from Helen Mayberg's work in 2005, where she used PET scan imaging to show that there was a decrease in prefrontal cortex neural activity. And in response to deep brain stimulation, there was a reversal of these of the activity that contributed to the alleviation of the depressive-like phenotypes. And overall, from these past imaging studies, it showed that there is hypofrontality that's associated with the negative symptoms in major depressive disorder. And Ron Duman further described that major depressive disorder patients, they reveal a reduction in prefrontal cortex neural activity, as well as PFC volume and dendritic spines. So what about the downstream circuits that are implicated in depression? So we looked to Melissa Warden's work in 2012, done at a Carl Desross lab, where she used optogenetics to selectively photostimulate these prefrontal cortex to dorsal raphe nucleus neurons. And she showed that in response to this stimulation, that there was an increase in kick frequency using the 4-swim task. And this is indicative of a promotion of motivation and also active coping. What was interesting was that when she stimulated a different pathway, for instance, the prefrontal cortex to the lateral habenula pathway, that this actually suppressed motivation and active coping. So there was a decrease in kick frequency within the 4-swim task, showing that there is these diametrically opposed prefrontal cortex projecting pathways that are responsible, or that could be responsible for depressive-like phenotypes. So our hypothesis was that there's a dysregulation in prefrontal cortex valence-specific excitatory projection neurons that induces anedonia, right? And our rationale is that a reduction in reward processing projecting neurons, accompanied with an aberrant increase in the aversive processing projecting neurons, will contribute to these depressive-like phenotypes. So I'll introduce you to what I'm currently doing during my postdoctoral fellowship in Dr. K. Tai's laboratory, which is investigating the effects of anedonia. And the first thing that we wanted to do was we wanted to validate a stress-induced protocol to induce anedonia. So as previously described earlier, anedonia is a core feature in both major depressive disorder as well as schizophrenia. And what is anedonia? This is the inability to experience pleasure or hedonic feeling. And it could be subdivided into these two different dimensions, such as hedonic anedonia, which is going to describe the reduction of liking or consummatory pleasure, or motivational anedonia, which will describe the reduction in wanting, desire, motivation, and anticipation. And this could be further categorized for simplistic purposes, such as physical anedonia, which will be a reduction in sensory pleasure, or social anedonia, which will describe the decrease in pleasure in social situations. So we'll use this model for the duration of the talk. So how are we going to implement anedonia? We elected to use the learned helplessness paradigm, which describes the interference of an adaptive response induced by an aversive stimulus. This was done by Seligman and Mayer in 1967 and further described in 1972. So with the learned helplessness paradigm, this gives an uncontrollable amount of stress to a subject that causes this subject to give up or to become helpless. So in order for us to do this within a rodent mouse model, we elected to use a shuttle box where we placed our subject mouse within one of the chambers. And we gave it 3 seconds of inescapable electrical shocks for 75 trials for 2 straight days. And this is the inescapable stress group. And within another group, the naive group, we would place our subject mouse in one of the chambers, and they received 0 shocks during these first 2 days. And on day 3, this is the testing phase. So we would reintroduce these mice into the shuttle box, this time with the door open, and we expose them to 10 seconds of escapable electrical shocks for 30 trials. And from there, we were able to delineate from this inescapable stress group, helplessness versus a resilient group. And the way that we determined this was by the amount of escape failures as well as the escape latencies in comparison to this naive group. So what do our results show? So we were able to threshold based off of the naive group, and we're able to threshold at the escape latency as well as the escape failures to determine this one quadrant shown here in red. And these are our helpless mice. As you can see, you get this distributed population that's widely variable in comparison to the resilient group. And from this inescapable stress group, we show that 43.5% of mice were considered helpless versus the 56.5% that were considered resilient. And if you look at the behavioral profile of these mice within the shuttle box, you can see that the helpless mice was significantly greater than both the resilient and the control group. And it was actually encouraging to see that the resilient mice shared this similar behavioral profile within the shuttle box to the control group. And as a side project, we also wanted to understand the effects of antidepressants on helpless behavior. And our hypothesis was that there are differential effects of NMDA receptor antagonist antidepressant treatment on these specific medial prefrontal cortex populations that would alleviate these depression-like phenotypes. And our rationale is that ketamine acts on the prefrontal cortex and at low doses will cause these antidepressant-like effects. So what is ketamine? Ketamine is a non-competitive NMDA receptor antagonist, right? And it binds to the PCP site of your NMDA receptors, and at low dosage, it will cause a state of euphoria, sensory distortions, and as well as feelings of empathy. However, at high dosage, you get these dissociative effects such as hallucinations, intoxication, as well as frightening experience. And one of the key problems with ketamine is that it can be used as a drug of abuse. However, it is technically a dissociative anesthetic. And if you compare it to your typical antidepressants, such as SSRIs and SNRIs, which have been shown to be highly effective, however, there's 10 to 30% of population that fail to respond to this treatment. So this is the treatment-resistant population where ketamine at one low dose would cause the alleviation of these symptoms for the treatment-resistant population. And it typically happens within hours, and it targets a specific system. It targets the glutamatergic system in comparison to your typical antidepressants, which will target the serotonergic and dopaminergic system, and it usually takes weeks for it to take effect. And ketamine has been approved in 2019, FDA approved, and it's only for fast-acting treatment for patients who have failed to find relief with at least 2 of the antidepressants. So there were some few follow-up studies that looked at what is ketamine doing to cause these antidepressant-like features. And one study, which was further described by Anna Baylor in 2019, she was describing Connor Liston's work in Ithaca. And what they showed was from 2-photon microscopy is that in response to stress, you get a reduction of these synaptic spines within the prefrontal cortex. And at one low dose of ketamine administration, then you get this synaptic formation of those dendritic spines. So you get new synapses within the pyramidal neurons of the prefrontal cortex that's responsible for the alleviation of those depressive-like phenotypes. So we wanted to test this on our helpless mice. So remember, at a low dosage, you get this state of euphoria and sensory distortions, as well as feelings of empathy. And what we did was we ran our mice within the shuttle box. We were able to discern whether they were helpless versus resilient. And we gave our mice a 10 milligram per kilogram IP injection of ketamine and recorded the escape failures as well as the escape latencies. And we showed that in response to this ketamine administration, which is 24 hours later afterwards, that there was a dramatic decrease in the latency to escape, as well as a dramatic decrease in the escape failures, indicating that these helpless mice were not helpless anymore. So in summary, for part one, learned helplessness protocol induced helpless and resilient mice, and ketamine administration actually eliminated this behavior. Now we want to follow this up by understanding the specific dynamics of ketamine administration and looking at those specific prefrontal cortex circuits, projecting circuits that would be involved in the alleviation of this helpless behavior. We next want to test the effects of anedonia by looking at one of our models that we were earlier describing in relation to reward consumption as well as social behavior. So what are the effects on reward consumption and social behavior? So we want to use this similar model to describe that: physical anedonia, which will be the reduction in sensory pleasure, versus social anedonia, which would be the reduction in social interactions, or that decreased feeling in social interactions. So in order for us to investigate this, we created this behavioral paradigm where we could test this. So we established, we started this sucrose preference test, where this is a 2-bottle choice task where we expose our mice to sucrose versus water, and typically a control mouse would prefer sucrose over water, right? And we also ran a 3-chamber sociability task where we introduced a novel mouse within the chamber, and we saw the interaction between our subject mouse versus this novel mouse as a measure of sociability as well as social exploration. We then placed our subject mice within the learned helplessness box, and we were able to discern whether they were helpless versus resilient, and then we scored them afterwards. So what do our initial results show? So from our naive mouse group, we were able to score the change from baseline to that testing day 3. And we saw that within the naive group that there were no significant changes in the sucrose preference test, okay? So they still preferred to consume sucrose over water. Now, when we introduced the helpless mice, this is where we started to see a significant decrease in the sucrose preference test. When we looked at change from baseline to testing day 3, we saw a significant decrease more specifically, and this was indicating anhedonic behavior, and more specifically, we're describing this as physical anhedonic behavior. And interestingly, the resilient mice showed a similar profile to the naive mouse group. Now, when we ran the 3-chamber sociability task, where we place our subject mouse within one chamber and we allowed it to interact with either this juvenile mouse or an empty enclosure, and we recorded the change from baseline to testing day, we saw that there were no significant changes within this mouse group, indicating that they still had a social preference for interacting with the juvenile mouse. Now, when we looked at the helpless mouse, we saw a significant decrease in the social preference from baseline to testing day. So, and these mice typically would prefer to interact with this empty enclosure versus this novel mouse. So this is indicative of a reduction of sociability as well as social exploration. And you compare it to the resilient group, which showed no significant changes and was similar to the naive group. So this was encouraging to see that these helpless mice were different from this resilient population. And to further describe our model of physical anedonia versus social anedonia, we wanted to see if there was a correlation between that decrease that we saw within the sucrose preference test and the 3-chamber sociability task. And this is what we did here. And we did see this significant correlation in both of these parameters, just further validating that we can continue with this model as a measure of anedonia. And we compared this to the resilient mouse group, where there was no significant correlation in either the sucrose preference test or the social preference test. So this is just further validating that these helpless mice did show not only a reduction in physical anedonia, but also social anedonia. So since we got these results, we wanted to further characterize the severity of these these anhedonic mice. And we went to the DSM, which described the depression in a gradient, for instance, severe depression, moderate depression, and mild depression. And this is what we want to do for the next steps in classifying helplessness within our mice. We want to create this phenotypic classification based off of these initial 2 different dimensions that we're describing as now, such as sucrose intake and social interaction, just so we could create a degree or establish a degree of helplessness within these mice. So this is definitely work that's ongoing. So in summary, for part 2, these helpless mice, they displayed a reduction in sucrose preference, indicating anhedonic behavior, but they also showed that there was a decrease in social preference. And the resilient mice displayed no change in sucrose preference or sociability. So lastly, in our next step, we want to investigate the neuronal populations and cortical circuits that are implicated in anedonia. And a lot of this work is still ongoing. We're still analyzing most of this right now, and we have a lot of future experiments coming up. But the goal is to examine the effects of the prefrontal cortex valence-specific neuronal populations in response to this stress-induced anedonia. So I'll take you through our paradigm that we came up with, where we will first start off with stereotaxic surgery, where we're injecting a genetically encoded calcium indicator as well as channel rhodopsin in select regions to optogenetically manipulate these specific medial prefrontal cortex circuits. But we also are establishing these baseline recordings from sucrose preference tests as well as the 3-chamber sociability task. And we're electing to use in vivo 2-photon calcium imaging to monitor neuronal activity over time. So this is one of the benefits of using 2-photon microscopy is not only the cellular resolution, but we want to track the same neurons over time. And in order for us to identify these specific neuronal populations, we designed this Pavlovian conditioning protocol underneath the 2-photon, where we pair a conditioned stimulus with a reward presentation, which is going to be our sucrose presentation, versus an association, a different tone association with an aversive stimulus, which is going to be our air puff in this case. And we did this for 60 trials. So after we identify these specific reward positive cells versus aversive positive cells, we then place our mice back in the shuttle box, and we could discern which mouse was helpless versus resilient, and then place them back underneath the 2-photon microscope. And we could now track these same cells and understand if there was any neuronal ensemble recruitment or elimination, or synaptic plasticity changes that were occurring afterwards in response to this learned helplessness procedure. And then we'll further validate this with sucrose preference tests as well as the 3-chamber sociability task. So first, I want to take you through our setup, which took a very long time to build. And this was primarily engineered by a previous postdoc in the lab, Dr. Kyle Fisher, who really designed and put a lot of work into building this entire setup. And this was further, as well as the entire team 2P. And this was further implemented by a research tech in the laboratory, Jeremy Delahanty. And you could see our mouse is sitting very comfortable in its mouse holder. And this is a head fixed apparatus that's placing our mouse right underneath the 20x objective, which is going into our 2-photon microscope. And also you could see where the spout is that's responsible for sucrose delivery, as well as a vacuum to suck up the sucrose if the mouse does not lick. And also you have our speaker for to elicit the different tone associations. I have a camera on the opposite side for to get the facial recognition features. And also on the other side, what you're not seeing is there's a needle to elicit this compressed air to as an aversive stimulus for the mouse. And this is just a video of what our mice look like underneath the 2 photon. You can see this very high resolution camera is going to be used for facial recognition, as well as this air puff where we're able to detect blink detection even pre-wincing before an air puff is elicited. And this is an example of what our imaging looks like, where we injected the genetically encoded calcium indicator GCAM7F within the prefrontal cortex, more specifically the prelimbic region. We have our Grin lens implant to monitor these calcium transients. And we're monitoring these calcium transients and using it as a proxy for neuronal activity. So this is just this is typically acquired at 30 frames per second. This video was dramatically sped up just for visualization. And during this imaging session, we're eliciting these different tone associations that is responsible for either reward delivery versus the air puff, which is going to be an aversive stimulus. And the overall goal is to identify which cell populations are activated during this reward stimulus versus the aversive stimulus. And then once we place them back under, or place them into the learned helplessness box and decide which mouse is helpless versus resilient, then we could understand the neuronal ensemble changes that are occurring within these specific populations and use optogenetic stimulations to specifically photoactivate the ones that are affected. So I want to also take you through this Pavlovian conditioning protocol that we customized and designed underneath the 2 photon, where we trained our mice for 7 days in head fixed boxes, and we were able to establish these baseline recordings. So for instance, you have this conditioned stimulus where there's a tone that's associated with this reward, which is going to be 30% sucrose, versus another conditioned stimulus that has a different tone that's associated with the aversive stimuli, and that's going to be the air puff. And once we establish this baseline recordings, we expose our mice to the learned helplessness box, place them back underneath the 2 photon, and we run the similar 60 trials. Then following that, we are injecting our mice with this low administration of ketamine, and then we place them back underneath the 2 photon to see if there are any changes in not only behavior, but also neural activity. And the first thing I want to show you is the behavioral outputs from our helpless mice. Now, I'm showing you the response from these 4 helpless mice that we already discovered post hoc, whether they were helpless or not. But first, I'll show you the lick probability. So this is just a behavioral score, and what you're seeing within this shaded region is the anticipatory licking. So these mice did learn this task following training. And then after the anticipatory licking and the US is presented, you could see that the mice continue to lick afterwards. Now, what was interesting was that once we put them back underneath or put them through the learned helplessness protocol and then put them back underneath the 2 photon, these mice did continue to lick, which is great to see that they continue to learn this task. But it also appears that they even licked a little bit more than they did during the baseline recordings, and they licked even further after the US was presented. And even following ketamine administration, they also continued to lick. So ketamine did not alter their learning abilities, and they also performed very well following that, following this procedure. So after we averaged the Z-score, we could see that there were that these mice did learn this task following the learned helplessness protocol, and it was encouraging to see that it did not affect them. But one of the things that we're still in the process of analyzing is the neuroactivity within these 4 helpless mice, and even afterwards, the changes in ketamine administration. So what we can get from this data is, are we able to, one, establish and identify a biomarker that could predict this helpless behavior, since we are not only getting the baseline recordings from our behavior, but we're also getting the baseline recordings from neural activity. Can we predict helpless behavior before? And also, can we predict which mice are responsive or more susceptible to ketamine administration? So in summary, for part 3, these mice undergoing this learned helplessness protocol, it required this conditioned tone sucrose association as well as the tone air puff association. And ketamine administration has no effects on this conditioned tone sucrose association as well as the tone air puff associations. So for future studies, we are using optogenetic manipulations to target these prefrontal cortex inputs, more specifically the dopaminergic inputs from the VTA. And we're also planning to use computational-based behavioral tracking systems such as Alpha Tracker to track the helpless behavior. And following that, we're going to be using ex vivo electrophysiology to isolate these specific synaptic proteins within the specific prefrontal cortex projection defined neurons. This is the prefrontal cortex projection defined neurons to the lateral habenula, as well as the PFC projection defined neurons to the dorsirapine nucleus. And with that, I would like to thank everyone in the Thai laboratory, especially my amazing and superhuman mentor, Dr. K. Thai, who's been an amazing mentor and coach, as well as my team members, which seem to be growing every single day, and everyone in the Thai laboratory, especially the entire Team 2P. And I want to thank my funding source, from the NIH, the F99K00. I received this grant at Drexel University in Dr. Wenjun Gao's laboratory. So it's been a huge support. Also want to thank UC San Diego, for where I teach cellular neurobiology over the summer. And I would be remiss if I did not bring up the Black Association at Salk. In fact, I want to bring this up now. So the Black Association at Salk was founded by myself and my partner in crime, Jasmine Ravena, who is a PhD student in Rusty Gage's laboratory. And one of the things that we noticed last year was there's an underrepresentation at Salk. So there's specifically from Black and African individuals, who make up just 1% of the population at Salk. And there were no Black groups or organizations on campus. And more so, there was lack of awareness of racial issues just in academia overall. So knowing this, we partnered with the Office of Equity and Inclusion, and specifically manager Lauren Mitchell, who's been a warrior for us the entire time. And we're able to create this new affinity group called the BAS. And the BAS, it acts as a service group. So we are able to create specific events to not only educate the entire Salk campus, but also outside of the campus by throwing these specific events. We're also a professional group. So we offer any type of career opportunities that come our way, where we're able to distribute this information out. And we're also a social group where we throw specific events on campus and even outside of campus. And one of the major things that I got was, how do we form this group? And what I did earlier on from the beginning was first reach out to any of the Black individuals that are on campus to form this core group, to first bring us together as a small group. And from there, Jasmine was able to reach out to anyone who was not a Black individual to become a supporter for us. And from there, we were able to establish this very large group at Salk. And the membership is open to all employees and all students, That's administration, research staff, support staff, graduate students, postdoctoral fellows, and a lot of support from faculty. But mainly, we got a lot of support from SALT. And what I mean support is we got monetary support as well. So we were able to secure funding throughout the year to throw these specific events. And we could also support these other Black-owned businesses and even support other organizations outside of the SALT. So I'll take you through a few of our events that we've thrown. And one of the major events that we threw earlier this year was Black History Month, right? So we designed these specific events throughout the entire month, such as Black Jeopardy. We had a SALT discussion circle where we discussed this highly acclaimed book, Stamped. We had coffee with Dr. Chrissy Towers, who is the first Black faculty member hired at the Salk Institute. We invited prominent professors such as Dr. Gentry Patrick from UCSD to give us a fireside talk. We also had yoga and Black mental health events. We also have film marathons. So we were highlighting specific actors and actresses and filmmakers to really help the entire Salk campus understand Black culture just in general, and especially from in great films like this. And we also were able to start our own Black library. And by doing this, we partnered with the Salk Library, and we were able to send over a list of any of the Black authors that we came up with, and we had them on display during Black History Month. But we also have all these books now integrated within the Salk Library. And earlier this year, we were able to set up the very first Juneteenth event on campus to celebrate the freedom of African Americans. And during this event, we were able to educate by having all different type of articles that we could tap into from our phone. We also had music at this event, and we invited specific Black-owned businesses to come on campus and share what they were selling. So this was a huge event where this was open to the entire campus, and we had a lot of people come out and support this event. This is one of the first events that was done by the event staff during the pandemic. And also, we were able to support these Black-owned restaurants. So what we did was we created a list of all the Black-owned restaurants within the San Diego area, and we had these on display so people could go there and not only purchase everything, but if they wanted to donate to these Black-owned restaurants, we could do that. And also, we became official sponsors of prominent organizations such as Black in Neuro and Black in Cancer. And this is one of the major things of getting funding and budgeting from the Salk that allowed us to actually donate and sponsor to these other organizations. So that was huge for us. And our future plans are to partner with other Black organizations within the area, such as Scripps and UCSD. And we're also going to launch our social media account, so look out for that in January, February time. And the goal is to also create this model for other research institutes to adopt. And one of the key questions that I get from people outside of Salk is, how did you form this group? And I obviously cannot do this by myself as a lowly postdoc. So I got this support from all the BAS supporters as well as the core members. And even outside of the BAS, we got a lot of support from, like I said, faculty members, and just getting that monetary support. And everyone like creating this entire new culture at Salk, which basically allowed us to pull these events off. So this has been very encouraging and very excited about the future of the BAS. So with that, I will take any questions.
Wonderful. Thank you so much, Austin. it was just incredible to hear about your work. Blown away, but not surprised. You've always been just such a star in the lab. So it's really great to see how your work has progressed. And if you weren't already on most people's watch list, you definitely will be after this. I'm sure people will be looking forward to see how your future directions pan out and really kind of digging into the underlying mechanisms of some really cool behavioral phenomena that you were able to demonstrate. It was really cool too that you got to talk about your work with the Black Association. You all have done so much in such a short time. And so I'm sure that this will serve as a blueprint for people looking to kind of start that type of work at other institutions. So thank you so much for sharing that too. We have about 13 minutes for Q&A. We have a lot of really great questions in the chat, but yeah, if you want to go ahead, if you haven't been able to ask your question, if you want to drop it in Zoom's Q&A box, I'll go ahead and ask them live. One question we got was from Danielle Jones. They asked, what makes the resilient mice resilient? A tricky question. I'm sure that's what you're trying to figure out. But if maybe you could speculate on that, what you think may be involved, whether it's certain genes or social features that may be important for kind of answering questions about what may underlie human resilience.
Yeah, so that's a great question. I've been getting that a lot, is like people are pretty like real interested on the resilient mice. Like, how are they resilient? How are they doing this? And I think one of the things that we're going to investigate is the coping mechanisms of these resilient mice. So it appears that although they're getting through the task, we think that there's other behavioral coping mechanisms that we're not seeing just from those two different dimensions that we would be able to look at through specific tracking systems that we plan to do a little bit later on, that allows them to be resilient. Now, we don't think that even though they are resilient, we don't think that the neural activity is going to mimic the exact same thing as the control mice. We actually think that there will be alterations that we will see during our 2-photon experiments that will help explain a little bit more on why they would be resilient. I have not thought about the whether there was genetic alterations that or even synaptic alterations that's going on and that would make them resilient or not. So that will also be in the works. But the first thing is to see if there's any neural activity changes within the resilient mouse group, as well as some coping behaviors that we're just not seeing right now that would have them like make it through the initial task. But that's a really good question. I'm really interested in figuring out what's going on with the resilient mice and how they are getting through these tasks. But I don't think they're relatively normal. I mean, if you're getting shocked that much, I mean, something has to be going on somewhere. So we just have to figure out where.
Okay, thank you. We had another question from Mike McDonald. They said, awesome job, and said it was really interesting to see the positive relationship between physical and social anedonia. So could you speculate on whether they might utilize the same neural circuit?
Well, hello, Mike. Thank you for tuning in. Yeah, so that's actually a really good question. If it's the same circuits that would be involved in both physical and social anedonia, and I don't imagine it to be the exact same. And I would say that I have not seen anything related towards social anedonia as relates to the PFC, the lateral habenula pathway, just yet. So we may have to look at specific circuits for, let's say, that would incorporate the ventral hippocampus that could be involved in sociability. The social aspect actually was one of the things that we added on to, and we didn't think that we would see any changes within the helpless mice. But we have not really sort of explored how if those circuits would be different or not. I would not imagine, I couldn't imagine that for both the physical anhedonia and social anhedonia, it would be routed through the same pathway. So it's actually a really, really good question on if there is a difference in those two different pathways, which we'll probably have to do some thinking about it, some probably in vivo imaging where they would not be head fixed to figure that out. But that's a really good question. And yeah, I would just say I would be surprised if it was routed through the same pathway. But thanks for asking that, Mike.
Yeah, quick follow-up to that. We have a question in the chat about whether you have or plan to use something that could be freely moving, like fiber photometry, to look at the same circuits.
Yeah, I think that's definitely going to be in the works, and I think using fiber photometry, especially for a social interaction task, will like really hone in on that specific question, because, or I mean, we are throwing out a lot of ideas on how we could still implement a social interaction underneath the two photon within this head fix model. We have thrown out many specific procedures on how to do this, which it's not entirely impossible for us to do. It would take a lot of reconstruction underneath the two photon, but we will probably end up doing the fiber photometry method to look at freely moving. So that will definitely probably be in the works. So thank you for asking that.
Great, thank you. We have so many questions, so unfortunately we probably won't get to all of them, but I'll definitely send them all to Austin so he can respond to them, maybe on Twitter or whatever works best for you. Our next question comes from David Weinshanker, and they said, great talk, Austin. They asked if you could clarify what you meant in your future directions about using electrophysiology to identify synaptic proteins.
Yeah, thank you, David, for attending and asking that question. Yeah, so we want to highlight and really want to focus on the specific proteins, more so the dopaminergic receptors on those prefrontal cortex neurons. So we want to first label the prefrontal cortex projecting neurons using probably retrograde labeling, whether it be CTB or retrobeads. So we want to first label both the prefrontal cortex to dorsal wrapping nucleus neurons as well as prefrontal cortex to lateral habenula neurons. And if we are photostimulating these dopaminergic inputs, then we want to focus on the DRD1 receptors in those selective neurons, as well as the NMDA receptors on those neurons. So I have another bold hypothesis out there. The DRD1 receptors are influencing those NMDA receptors, and more specifically the NR2B containing NMDA receptors on those specific populations. So we could photostimulate that and then record the and use a specific drug to either as an antagonist for the DRD1 receptors and record the NMDA receptor excitatory postsynaptic currents from those specific population of neurons. Then we could hone in on those on the synaptic abilities of the glutamatergic projecting neurons. So those are the specific proteins that we would want to focus on within the prefrontal cortex as of now.
Awesome. Another question we had from an anonymous attendee, they asked they commented on the fact that some studies in rats have shown that repeated low dosage of ketamine can induce a schizophrenia-like phenotype. Have you seen that in any of your work, and have you taken a closer look at what your ketamine administration is doing? Are you seeing anything like that in your mice?
Thanks for that question, anonymous. And I think that we will have to further sort of validate the ketamine administration by looking at first like head fix or head twitching of these mice to see if they are experiencing any psychotic symptoms. And I think that's one of the sort of established paradigms for looking at psychosis in a rodent model. So that is one of the things that we will have to do, is sort of make sure that we're validating that these mice are not undergoing psychosis following their ketamine administration. I will say that behaviorally afterwards, they are overall less anxious, and even they performed very well during during the Pavlovian conditioning protocol. So the fact that they appear to be cognitively there, and also there's less anxiety afterwards, that was pretty encouraging. But we do have to sort of validate that these mice are not going under psychosis. So that's probably the next step, is to do the head twitching procedure on these mice.
Awesome, thank you. We have time for about 2 more questions. So I'm gonna ask a multi-part science question, and then we're gonna ask one career question. We have some of those in the chat too. So this comes from Tara, and Tara asked, well, they said it's a very creative behavior assay, which definitely was very cool. They were curious if your animals are group housed or singly housed. If group housed, is there some variability within a cage, or do they tend to take on the characteristics of their cage mates? And they're curious how the emotional state of the cage mates might affect the behavior of the mice that you're testing.
Oh, yeah, good question. So these mice were all group housed because we know that in response to social isolation that they would be a lot more anxious. And one of the things that we did record, which we just haven't finished analyzing yet, is we're recording the home cage sociability within these learned helplessness mice following their group housing. So, and we're using Alpha Tracker computational behavioral analysis to track this to figure out if there is any social dominance within their home cage from a helpless mouse versus, let's say, one of the resilient mice. And that's one of the things that we do want to sort of monitor over time. It was great that we did the 3-chamber sociability task, but seeing how they are in their natural environment with their siblings to see like if there was any changes in the social interaction within those mice, that's going to be like huge for us. Furthermore, just to further describe whether not only the severity of the anhedonic mice, but we also want to see if there is a bully mouse in there, and if that like alters the mouse's behavior even afterwards. So that is one of the things that we did record. We just have to further analyze that and see what we get from it.
Thank you so much. We had a couple career questions that are kind of from different or touch on different arenas. So I'll just kind of try to put one together. Being where you are now, you know, you've been incredibly successful both at the graduate and postdoc level. So like, what's one piece of advice you would give to someone who is just getting started, to say they're thinking about like applying to graduate programs, that you wish you would have known as you were getting started, now having the hindsight that you do?
Oh, well, yeah, I wish I would have known a lot. Yeah, so I mean, I will, that's a really, really tough question. I'm going back to like when I first started off doing science, and even like, you know, seeing like how far I got as of right now. I will say that some good career advice would be to, one, do something that you're definitely like passionate about. So I was very passionate about science and research in general. And as I was going throughout my career, I started to really understand what I wanted to focus on for future careers. So when I was it was when I was at North Carolina Central. One of the only things that I really liked was biology and biology anatomy, and but I didn't really like discover anything like really after that in terms of like interest. And then when I was a master's student, I randomly got put on a neuroscience project. I was working with postdoctoral fellow Dr. Vernon Ruffin, and we were the only ones doing like neuroscience within this pH regulation lab. And I really fell in love with it, with neuroscience at that point. And I was like, oh, this is a really, really cool field. So that was like more motivation to continue with that. And then even after my master's, I was like, all right, so what about like neuroscience do I want to like continue with? And I really wanted to focus on neuropsychiatric disorders. So that's why I applied to Drexel University, and specifically wanted to work for Dr. Wen-Jen Gao, because he was focused on schizophrenia and he was using all the techniques that I wanted to use. So that was like another motivating drive to get to where I'm at, even during my postdoctoral fellowship. And as I was doing a lot of my interviews and coming up with what do I want to do for a career, I was thankful to when interview with Dr. K. Tai, and we were able to come up with a project that we both were really, really interested in. And that was like another further motivation to say like, okay, I want to continue with this path because these are the projects that I'm really interested in. So like, that's just one, I guess, piece of advice is to do something that you're definitely passionate about. And that will like give you like some more motivation to continue to get to like each level. But there's also like I said, there's tons of other things that I'm probably not thinking about right now and that I wish I would have known like early on. So I was just figuring out life as it's going. Still am.
Aren't we all? That's that's a great way to close. So there are 10 to 15 more great questions in the chat. I've saved them all, so I'll send them to you afterwards. So you can answer them on Twitter. People can also, I encourage them to reach out to you if they have more questions as well. If you want to drop your Twitter handle in the chat so people can find you.
And you can also email me as well.
Yeah, if you're comfortable, drop your email as well. So I just want to thank you for such an incredible talk. Like I said, it was just really, really cool work, and it's been great to see how your career has progressed, and I'm so excited to see what you do next. I know it'll be great. And the Black Association is such incredible addition to Salk Institute, and it sounds like it can definitely serve as a blueprint for people looking to do similar work at other places. If you want to share any tidbits you learned on social media, remember to do so using the hashtag #BeenSeminar-series. I definitely encourage you to follow Austin so you can keep up with his work and his service. I want to thank the co-organizers on the seminar series committee, Ashley and Paige, for their help in organizing this series. We have one more seminar coming up on the first Friday of January, and then we'll take a bit of a break before we get back to our next season. I want to thank Elena Dominguez for her help on the technical side of things, and then the whole just Black and Neuro team for everything that they do. And then I just want to thank everyone again who joined us for coming. It's been really great to see you all here virtually. It's been great to see your questions. You were such a great audience, and hope to see you at the next one. All right, thank you.
Thank you.
Yeah. We'll still be live on YouTube for a moment.